Welcome to Module 6 of Cells, Tissues and Regulation. In Topic 6.2, we will learn about renal function and fluid balance. During the course of this video, you can open your workbooks to Module 6 and try and answer as many questions as you can from Topic 6.2. The learning objectives for this topic are to understand how much primary filtrate is produced per day and by what mechanism, to describe the role of the proximal convoluted tubule in resorption of water and other solutes, detailing in particular the uptake of sodium chloride and glucose, to understand the process of secondary active transport of glucose, as described earlier in the course in the GI tract, but today in regards to kidney function and diabetes, to understand that amino acids are also recaptured from the primary filtrate by secondary active transports. And also with regard to the loop of Henle, we need to understand that a very high osmotic pressure is generated in the medulla of the kidney using these structures. And we want to relate that high medullary osmolarity to the role of anti-diuretic hormone, which, by stimulating the insertion of aquaporins into the wall of the collecting ducts, causes the production of a concentrated urine. We want to be able to describe how aldosterone affects the handling of sodium chloride in the distal tubule, and how this can also influence water balance. And finally, we want to understand that kidney tissues release renin in response to a falling blood pressure. The kidneys are one of the most important organ systems in our body, and are usually underappreciated until they fail and the body fluids become contaminated. The kidneys maintain the body's internal environment by regulating several key functions. The renal endocrine tissues release several hormones: renin, that controls blood pressure, and erythropoietin, which is known to stimulate red cell production. They also release hormones that play a part in the regulation of blood calcium and glucose levels. We will learn about all of these functions in different subtopics during Module 6. The kidneys also respond to hormones and control the total water volume in the body, as well as the concentration of various solutes in that water. It is vital that the kidneys regulate the concentrations of different ions in the extracellular fluid, because even relatively small changes in the concentration of sodium ions or potassium ions can be fatal. The renal system also plays a crucial role in maintaining a healthy extracellular environment for all the cells in our body by excreting unwanted metabolic wastes and ensuring acid-base balance. The kidneys are also the primary excretory organ of the body, and without them, all of our body systems would crash. The kidneys are the most important component of the urinary system itself, as it is here that urine is produced by removing metabolic waste from the blood through a filtration-like mechanism. The urinary system also includes the ureters, which are a pair of tubes that transport urine from kidneys to the urinary bladder. Then there's the urinary bladder itself, which is a temporary storage reservoir for urine. And finally, there is the urethra, a tube that carries urine from the bladder to the exterior of the body. Introduction to renal physiology will be heavily simplified, and most of the principles that are going to be discussed in this topic have been previously studied in this course in Module 2, such as the sodium pumps, which are involved in the reabsorption of sodium from the filtrate in the kidneys. We will also discuss the facilitated and the secondary active transport mechanisms through which glucose and amino acids are reabsorbed from the filtrate, and the importance of osmotic gradients that direct the movement of water from the kidneys into the extracellular fluid. If you look at the basic anatomy of the kidney, these bean-shaped organs have an outer cortex and a deeper medulla. The renal medulla exhibits a cone-shaped tissue structure, and these are called renal pyramids. These pyramids appear striped because they are formed almost entirely from parallel bundles of microscopic collecting tubules and capillaries. The kidney is covered and protected by a fibrous layer called the renal capsule that surrounds the adipose tissue. Huge quantities of blood are filtered through the kidneys by the nephrons. These are the structural and the functional units of the kidney. Each kidney contains over 1 million of these tiny blood processing units, which carry out the processes that form urine. In addition, there are thousands of collecting ducts, each of which collect fluid from several different nephrons and deposits it into the renal pelvis. Almost all of the filtered content is reabsorbed and returned to the body. However, toxic wastes are excreted, and the final amount of water and salt retained or lost in the urine will depend on our daily body needs. When we look closely at the anatomical structure of the nephron, each nephron consists of a renal corpuscle and a renal tubule. Each renal corpuscle consists of a tuft of capillaries called the glomerulus, and a cup-shaped hollow structure known as a glomerular capsule, which is continuous with the renal tubule. The afferent arteriole brings oxygenated blood into the glomerulus. The endothelium of the glomerulus is extremely porous to allow for filtration to occur. This property allows large amounts of solute-rich but protein-free fluid to pass from the blood into the glomerular capsule. The fenestrations, or gaps in the capillaries of the glomerulus, allow solute particles such as sodium, potassium, calcium, and glucose to pass through them and enter the glomerular capsule through the filtration slit. This plasma-derived fluid, called ultrafiltrate, is the raw material that the renal tubule will process to form urine. One of the key points to remember here is that the primary filtrate collected in the glomerular capsule is very similar to the blood plasma, but without plasma proteins. Because of their large size, proteins cannot pass through the fenestrations of the capillaries. Proteins in urine can only be found in cases such as infection, kidney disease, and pregnancy. There are roughly 1 million nephrons per kidney. Together they produce 180 litres of filtrate per day. However, as mentioned previously, most of the solutes in water will be reabsorbed. The total plasma volume filters into the renal tubule once every 22 minutes. If not for tubular reabsorption, which quickly reabsorbs most of the water and solutes, we would drain all of our plasma as urine every 30 minutes. The tubular resorption is selective and begins as soon as the filtrate enters the proximal convoluted tubule. Most of the solutes are reabsorbed by the proximal convoluted tubule. In fact, all of the glucose and amino acids are reabsorbed here. As you can see from the blue and red arrows, the reabsorption happens via both active and passive transport mechanisms. The primary active transport moves sodium out of the tubule, while glucose, amino acids, and most of the other nutrients are carried by the secondary active transport mechanisms. Because the solutes are continuously pushed from the tubules of the nephron into the extracellular fluid of the medulla, a strong concentration gradient is created, following which the water from the tubules of the nephron diffuses into the extracellular fluid of the medulla by osmosis. Furthermore, ions such as potassium, urea, and chloride diffuse across the membrane of the tubules into the extracellular fluid by passive transport. By the time the filtrate moves from the proximal convoluted tubules into the nephron loop, almost 80% of the water and electrolytes have been reabsorbed, which then diffuses into the nearby capillaries. In the descending limb of the nephron loop, water moves out of the tubule by osmosis. Here, ions cannot escape from the tubule because the descending limb of the nephron loop is impermeable to everything other than water. In the ascending loop and the distal convoluted tubule, ions such as sodium chloride and potassium are moved by active transport. Filtrate from the distal convoluted tubule then flows into the collecting duct, where ions such as sodium, potassium, and chloride can be reabsorbed. The hormone aldosterone controls the sodium and potassium levels in the body, whereas ADH, or anti-diuretic hormone, regulates water reabsorption here. Sodium is the most abundant ion in the filtrate. Roughly 80% of the total energy used by the active transport is devoted to reabsorbing sodium from the tubules of the nephron into the interstitial fluid. The movement of sodium and other solutes from the proximal convoluted tubule into the interstitial fluid creates a strong osmotic gradient, which pulls water into the interstitial fluid through transmembrane proteins called aquaporins. As mentioned before, anti-diuretic hormone can control water reabsorption, by increasing the number of these aquaporins in the membrane of the collecting duct. Glucose from the proximal convoluted tubule is transported into the interstitial fluid by secondary active transport. The secondary active transport is driven indirectly by a concentration gradient created by primary active transport mechanisms. Because the sodium-potassium pump of the primary active transport mechanisms continuously pumps sodium out, it creates a low concentration gradient for sodium. In secondary active transport, substances such as glucose are pumped from a region of low concentration to a region of high concentration. This process must therefore require energy, because glucose molecules are transported against their concentration gradient. However, the energy that drives glucose across the membrane does not come directly from ATP. Rather, it comes from the low sodium concentration gradient within the cell, created using ATP by the sodium-potassium pump. As sodium diffuses back into the cell and across the membrane, it takes glucose with it, against its concentration gradient. Because ATP does not fuel the carrier proteins directly, this process is known as secondary active transport. Anti-diuretic hormone, as the name suggests, inhibits urine output, or diuresis. The interstitial fluid in the medulla of the kidney is highly osmotic, with an osmolarity of roughly 1200 milliosmoles. If given a chance, water inside of the nephron tubules would diffuse into the medulla by osmosis. ADH makes the cells of the collecting ducts more permeable to water by inserting aquaporins into the membrane. The amount of ADH determines the number of aquaporins, and thus the amount of water that is reabsorbed. When the body is overhydrated, the osmolarity of the interstitial fluid decreases because there is more water and less solutes. This reduces the ADH secretion from the hypothalamus and makes the collecting tubules less permeable to water as fewer aquaporins are present. ADH is also called vasopressin. This is because in large quantities it can increase blood pressure by increasing water reabsorption and also inducing vasoconstriction. Renin is an enzyme that is released by the renal tissues. The renin-angiotensin-aldosterone system is the body's main mechanism for increasing blood pressure. When decreased blood pressure and blood flow is detected by the special kidney cells, the juxtaglomerular apparatus in the kidneys release renin, which triggers several complex pathways that can increase blood pressure by increasing the blood volume and inducing vasoconstriction. We will revisit the renin-angiotensin-aldosterone system in blood pressure homeostasis. These are some of the basics of the relevant kidney physiology. In summary, you should understand and be able to explain the following: that sodium, glucose, and amino acids are reabsorbed from the proximal convoluted tubule by active transport. Because of the reabsorption of solutes, a strong concentration gradient is created in the medulla, which allows the osmotic uptake of water. Anti-diuretic hormone allows water to be reabsorbed from the collecting duct by inserting aquaporins, and therefore maintaining the water balance. Aldosterone plays a key role in the reabsorption of sodium. It primarily targets the distal convoluted tubule, and as a result, little or no sodium leaves the body. Increased levels of potassium, or hyperkalemia, trigger the release of aldosterone. And finally, low blood pressure stimulus and low blood flow to the kidneys trigger the release of renin, which stimulates several downstream mechanisms which increase blood pressure. In summary, from this video, we have learnt the kidney structure and function. We've learnt about nephrons and reabsorption of water and solutes, the role of renal tubules, anti-diuretic hormone and water retention, the action of renin, and about the kidneys in general and their homeostatic mechanisms.